Hi everyone, this is the video to the PDF that I just posted on how to complete the DNA or the genetic coding exercise. It's really it's pretty straightforward. I think once you see what's going on, I'm going to explain to you step by step how this goes, and I think you'll kind of enjoy it once you figure out how it's done. It's really not complicated though. Okay, just a simple transcription translation exercise, just an easy way for you guys to earn 10 points. Okay, so here we go. Okay, again, I just want to set your minds at ease. Don't complicate the issue. This is just a simple transcription translation exercise. Okay, don't freak out about it. It's pretty cool. So in this process, what you're doing is you're just regurgitating what you know about the central dogma. Remember that the central dogma, you're taking double-stranded DNA and you're transcribing it into RNA, specifically messenger RNA, which is single-stranded, as you see here, and then you're taking that message, that mature message, and you're translating it into protein. Okay, that's what we're doing here. And so you'll recall from chapter 15 that when you transcribe your messenger RNA, that's what you see here, the messenger RNA, you have to process it before you can translate it. Remember, transcription takes place in the nucleus, translation takes place in the cytoplasm. Transcription, you get the primary transcript, then you have to process it before it goes out of the nucleus to be translated. And the way you process it is you remove the introns, which we'll see in here in a second, and you pop on, we call it the 5' cap and the poly A tail. Poly A tail goes on the 3' end. It's just a bunch of adenine residues, chain of them. It can be 5, it can be 10, it can be 100, doesn't matter. And the 5' cap is a methylated GTP. Okay, that's the processing, and we can go ahead. Now, in all of this, you have to not remember the genetic code, but remember to reference the genetic code. I don't want you to memorize all of these amino acids and their codes and their codons and all that stuff. When you do this later on the exam, I'll give you a copy of the genetic code. At this point, refer to your notes, refer to my presentations, refer to your textbook, whatever you got there. This is from your textbook, copy of the genetic code. Now refer to it here as we get into the exercise. Okay, so we started out here. I'm going back to the original exercise, and I'm not using the same DNA. Okay, I redid it. I'm using a different one just to give you an idea, because I don't want you to don't want to give you all the answers. But it says below is a sample of double-stranded DNA. Identify the sense or the coding strand and the nonsense or the template strand by placing a plus and a minus respectively in the space of the left of each strand. So you're going to just put a plus or a minus over here to identify the coding strand and the template strand. Okay, the template strand is the nonsense strand. The coding strand is the sense strand. The template strand is the nonsense strand. Or we call it the template because that's where we're going to make our messenger RNA from. Okay, we call it the nonsense strand, and the coding strand. Okay, should I give it away? Of course, because that's what this is all about. So remember this is 5' to 3' in this direction, 3' to 5' in this direction, and this strand is complementary to that strand. Okay, remember, complementary base pairing. Now, what I did here, which might make it easier for you, you can do this on a separate piece of paper. I went through and remember, each amino acid is denoted by a codon. A codon is 3 letters. So, you see a bunch of letters in this strand up here, and it can look kind of daunting. So what I did, just to simplify it, I just broke it up 3 letters and put a dash between every 3 letters. Little dash, 3 letters, dash, and each one of these then is a codon, right? Because a codon is 3 letters. So just to make it easier on your eyes and ultimately on your brain, just break it up into individual codons. I did that with both strands. And then we're going to identify the coding and the template strand by putting a plus by the coding strand. The coding strand is the 5' to 3' strand. That makes the template strand, or the nonsense strand, or the anti-sense strand, because it's anti to the plus strand, is going to be the 3' to 5' strand. So this is the coding strand, and this is the strand you're going to use to make your messenger RNA from. This is the template strand. You can use this as a template to make your message. Remember that in DNA, A is a designation for adenine, T for thymine, C for cytosine, and G for guanine, okay? I'm just going to move this to the top of the page. And here we go. Now the next, okay, so this was your first instruction. So your next instruction says, in the space below, transcribe a messenger RNA molecule from the template strand above. Template strand is the minus strand. Be sure to indicate the 5' and 3' ends of the primary transcript. Now, primary transcript, because it's not going to be modified before translation. This is just the raw transcript. You're going to use the template strand, the negative strand, the nonsense strand here, and you're going to make your messenger RNA. Now remember, these are the bases, nitrogenous bases for DNA, and you'll recall from chapter 15 that in RNA there is no T. So it's adenine, remember, uracil, cytosine, and guanine, okay? So what that means is when you transcribe your template into your message, you have to replace, because there are no T's in RNA, you have to replace every T with a U. All right? Or rather, you have to replace every A with a U, because A is complementary to T. So when we transcribe A, we can't transcribe it into T because there is no T. We have to use a U. So what I'm going to do down here in this space, I'm going to transcribe the message. So we're going to have in the transcription, in the message, we're going to have, this is GGG in the template, so we're going to have CCC down here, TTT in the TTC in the template, so we're going to have AAG down here, right? You're just doing the complement. Here we have TAC in the template, but we don't have T's in RNA. So this is going to be AUG down here in the RNA, and then we have CUC, GAU, AUA. Okay, just like that. That's what I'm going to do down here. And it said, don't forget to indicate the 5' to 3' direction, right? We copied this, or we transcribed the template, which is 3' to 5', which means down here it's going to be 5' to 3'. Now, what you will notice, what the astute person will notice, is that if the template strand is complementary to the coding strand, then the transcribed message must be complementary to the coding strand. And so you can do this by going one codon by one codon like that down here, or it's the same, right? 5' to 3', CCC, CCC, AAG, AAG, and the only difference is wherever you see a T, you're going to put a U. So ATG, AUG, CTC, CTC, CUC, GAT, GAU, just like that, all the way down. See? CCT, going to be CCU and at the end, okay? So it's the same thing. This is the coding strand without the T's. Replace the T's with U's. All right? This is your raw message. This is your primary messenger RNA, okay? We need to modify it before we can translate it. I'm going to move this up here. Now what I've done here is, okay, these, and now once again, each amino acid is 3 letters. We call that the codon. So this is the same thing that we ended up with in the previous slide, but now we have to modify it, and part of that modification is determining where the polypeptide starts and where it stops. And as you know now from chapter 15, that AUG is the only start codon in the whole genetic code. AUG, it codes for methionine, and that's important to remember, but it is the only start codon. So what you do in your exercise, your homework exercise, is you just go through that big chain from left to right, from 5' to 3'. Just go through that big long line of letters, and when you come to an AUG, that's where you know to start. I put a little green circle there just to remind you that's where we're going to start. And then you're going to read 3 letters at a time, 1 codon at a time, until you get to a stop codon. Now there are 3 stop codons in the genetic code, and your stop codon here could be any one of those 3. But in my example here, it's UAA. So you're going to read from the start until you get to a stop codon. In my case here, it's UAA, so I just put a little red circle there just to remind you that we're going to stop translating right there. Now, everything that is after the stop codon, well, I should say everything from the stop codon and after that, because the stop codon, remember, doesn't code for an amino acid. So when we process the message, we got to get rid of the stop too. So from the stop codon on, everything is nixed. Okay, that's all intron. The start codon codes for methionine, so we want to keep that, but everything prior to the start codon is intron, so we want to nix that. So what I've done here in the space below to draw the mature mRA, I get rid of the introns, get rid of the stop in the intron, and I just have the mature message there, and it looks like this. Okay, there's AUG, AUG, CUC, CUC, GAU, AUA, UAC, CGA. Okay, no tricks here. Now, the other thing we have to do to process this, according to the instruction, we have to add the methylated GTP cap and the poly A tail, the 5' cap and the 3' tail. So that's all I'm going to do here, okay? There's the 3' tail and the 5' cap. Now, the poly A tail is just a bunch of adenine residues. It could be 5, it could be 10, it could be 100, as I said before. I have 4 here just to give you the idea. Now also I've done these nice little red circles and I've linked them together, and when you do this you don't have to do that. You don't have to do that. I just did this to make it pretty for you, interesting to you visual learners. You can just put a circle there and put poly A tail, or you can put a circle there and put A. Okay, I don't care how you do it. Just signify that there is a poly a multi-adenine residue tail there, and on the other end that's a methionated GTP. Remember, GTP is guanosine triphosphate, just like ATP, adenosine triphosphate, but this is a G. It has a little methyl group on there. So that goes on the 5' head. You can put 5' head if you want, okay? 3' tail. You should say poly A tail though, but there's no convention here. Just let me know that you know what goes on either end. All right, I'm going to move this to the top. Okay, so it says in the space below, translate the mature message. This is the mature message now. We've gotten rid of the introns. We've gotten rid of the stop codon. We've capped it off and put a tail on it. Now this thing goes out of the nucleus. Remember, transcription takes place in the nucleus, translation takes place in the cytoplasm. This thing goes out of the cytoplasm, out into the cytoplasm. That's the reason we put these ends on, so the exonucleases don't chop this thing up, because this is an important protein. So we don't want it to get destroyed. That's why we put these things on there. Okay, but when we translate the message now, when we make the polypeptide and ultimately the proton, we don't need these things. All right, so it says in space below, translate the mature message that you drew above. Here it is. Be sure to indicate 5' and 3' ends, and indicate amino acid residues using their 3-letter designations. And then your instruction also says refer to the genetic code to complete this part, because you don't remember. You're not going to remember what the 3-letter designations are for these amino acids. Okay. So here's the genetic code that you're going to refer to. This is from your textbook, and the same image that I used in the chapter 15 lecture. And you can see that these are the amino acids. There's phenylalanine, leucine, isoleucine, threonine, proline, there's arginine, cysteine, tyrosine. Okay, you get the idea. Each one has a 3-letter designation, and that's here. It's the first 3 letters of the name of the amino acid. So phenylalanine is PHE, serine is SCR, proline is PRO, alanine is ALA. Okay, you get it. That's the 3-letter designation that when you translate your message, that's what you want to stick in there. All right, these are the codons. Now, and there's your methionine. That's the only start codon. There are your 3 stop codons, just for reference here, and they do not code for anything. Okay, so just to make this a little bit clearer, I'm putting up here the mature message that you made in the previous slide, okay, or made in the previous step. This is just the same message, 5' to 3', okay? So AUG, we know that codes for methionine. That's the start codon, but we know it codes for methionine. It's a start codon, but you can't get rid of it because it codes for an amino acid. So AUG, when we do our translation, instead of AUG there, we're gonna put MET. CUC, you gotta look. First letter is C, second letter is U, and there's CUC right there. It codes for leucine, LEU. There's GAU, first letter is G, second letter is A, GAU, which codes for aspartate. There's AUA, first letter is A, second letter is U, right? And third letter is A. There's AUA, codes for isoleucine. UGU, here's U, second letter is G, third letter is U. Cysteine. There's UAC. Here's U. Second letter is A. Third letter is C right there. Codes for tyrosine, TYR. And finally we have CGA. First letter C. Second letter G. It's over here. There's your G. Second letter G. Third letter is A. CGA. That codes for arginine, ARG. So those designations, those three-letter designations, are what you're going to put in the final step. Okay, and here it is again. So remember, in your mature message, you're going to get rid of the poly A tail, you're going to get rid of the GTP, and you're going to keep the 5' and the 3', and you just translate it right down the line, and that's what I'm going to do using the three-letter designations for the amino acids. Okay, not the three-letter designations, but what I wanted to do is to show you once again the mature without the GTP, and without the poly A. Here it is. And then I'm going to translate each of these codons into its appropriate amino acid. AUG, as you know, codes for methionine, MET. CUC, as we saw, codes for leucine, that's LEU. GAU for aspartate, not aspartame, that's the artificial sweetener, aspartate. That's ASP. AUA, that's isoleucine, ILE. UGU is cysteine, CYS. UAC is tyrosine, TYR. And CGA is arginine, that's ARG. Okay. I just did it like this because I'm a visual person and I like all the pretty colors, and so that's the way I did it. You don't have to do it like this. All right, you can just, in fact, you don't even have to make circles like this. You can just do 5' MET dash LEU dash ASP like that. And so yours will probably look something like this. There you go. 5' MET dash LEU dash ASP dash ILE dash CYS dash TYR dash ARG, and don't forget the 3', okay? And that's your final step. And that's how you transcribe and translate DNA to protein, okay? And that's the exercise. Hope you have fun with it, kind of like a little puzzle, I guess. And then you can do this. You can do this on paper if you want. Just make sure I know your steps. Shoot me a photo. You can download the PDF and do it with your ad text if you want, and upload it that way, however you want to do it. If you have already tried this out, don't worry about it. I've went ahead and opened it up. I've extended the due date until next week, December 6th. You have plenty of time to do this. And as always, if you have any questions, give me a shout.